Hi, everyone, and welcome back to the Smart Tea podcast, where we talk about the lives of scientists and innovators who shape the world. I'm Aarati.
And I'm her mom, Jyoti.
And Mom, I'm really curious if you've heard of the scientist we're going to talk about today. His name is Roger Tsien.
Tsien?
Tsien. Yes.
Hmm. It rings a bell, but I'm not able to place it. But
No?
I am
Okay.
pretty bad with names, you know that.
But you know what? This is exactly what this podcast is about, because I know for a fact that you have worked with his discovery or what he's known for literally every single day of your life.
This is becoming more and more intriguing by the minute.
So Roger Tsien, along with Osamu Shimomura and Martin Chalfie, jointly all won the Nobel Prize for their work on fluorescent proteins.
Oh, you are right. I do, and I have extensively used fluorescent proteins. It is such a beautiful way to track cell-cell interactions, things happening within the cell.
Mm-hmm.
And you can tag proteins with different colors
Yes.
so that you can then have beautiful pictures of what various proteins are doing with respect to each other, where are they located with respect to each other, what happens when something is added, and so then they change and align differently, do things differently. Wow, I did not know that they were responsible for it. This is going to be so much fun.
And I also thought, in order to add to the fun, we should play a game while we're going through this story. So every time I mention a Nobel Prize winner, we're going to take a shot or a sip of water, because this story is just littered with them. There are Nobel Prize laureates everywhere in this story.
This is fantastic. So, this is work from a very high level.
Oh, yes. Almost immediately, Roger is surrounded by amazing scientists and role models. And so he's basically set up for greatness from the beginning. We're going to be well hydrated at the end of this story.
That sounds good. I like being hydrated. Okay.
So we're starting with his amazing father. His father, Shui Chu Qian, was an aeronautical engineer, and he was serving in the Chinese Air Force when he met Roger's mother, Yi Ying Li, who was a nurse. They started a family there, and they had their first son named Yong Yu.
Hmm.
Then, during World War II, Shui Chu was ordered to go to the US as a liaison to try and get more aid for the Chinese military. But because there's a war going on, he had to zigzag all over the world in order to avoid enemy forces. And
Ah,
so by the time he landed in the US, the war was over. Japan had surrendered, and now Shui Chu is in the US. And he realized that since China was on the losing side of the war, if he went back there, it would be tough to build a life, because China would be trying to rebuild from a
yes,
war that they had lost.
that's right.
And so he instead made arrangements for his wife and his young son, Yongyu, to come and join him in the US.
Oh, I'm surprised that it happened. It seems to have happened fairly easily because at this point, I would have thought that the US would think of China as the losing side and therefore the
The
other
enemy,
basically, the enemy
yes.
exactly. And yet they allowed a Chinese family to be reunited.
Yes, so they were able to immigrate, but to your point, life was still a struggle for them exactly because of that. Shui Chu had a really hard time finding a job as an aeronautical engineer because he couldn't get the security clearances that he needed to work in that field.
Yeah, that actually makes a lot of sense. Wars leave behind a lot of mistrust, especially if they will be looking different. They seem to be different. I can see the human prejudice coming into play here.
Yes, very strongly. And so instead, in order to get by, he started a small import-export business, and then later started a engineering consulting company,
Hmm.
which, allowed them to kind of get by. They weren't super wealthy or anything,
Hmm.
but they were managing. The couple had two more children, a son who they named Yonglo, And then their third son, Yan Qian, was born on February 1st, 1952 in New York
Hmm.
City. And he's the star of our show today.
Okay. So Roger Tsien was actually born Yon Chen.
Yes.
Okay.
So around this time, their first child, Yong Yu, was
Hmm.
just starting school. And the
Hmm.
school officials told him he needed to pick an American name.
Ah,
Yeah.
okay. Oh, this would not fly today. Okay.
Yeah.
All
Yeah.
right. But a small point here. One of the things I have noticed as I've navigated my way in America for so many years is that a lot of Indians maintain their name. If they change it at all, it is extremely small change. So, for
Hmm.
example, somebody could be like Raj Kishore or something, and
Hmm.
they'll just shorten it to Raj
Yes.
or something like that. So
Yeah.
something,
Which is
something
what
which
you
is
do in America also anyway, like,
anyway, correct.
yeah, like Jennifer becomes Jen.
Exactly. However, in contrast, I always noticed that people who came in from China would change their name drastically. Exactly like you said, they would say Yong Chen or something like that, but call me Roger. And
Yes.
I would just think to myself, but where is the connection?
Yeah.
Like, that doesn't make any sense to me. So
Yes.
maybe this is it. Since the Chinese immigrants came in much earlier, they had been
Hmm.
conditioned to do this. Like, you come into America and you change your name because your name is not pronounceable to the rest of the Western world. So you do it like this. But Indians came a bit later, where there was more openness already building in America. People were like, hey, I appreciate your culture, I embrace it. Tell me your name. I'm going to work my hardest to pronounce it correctly. Just
Yeah.
tell me how to do it. And that's what I faced always. I would tell my name, and then people would say, tell me again how to say it. And then I would say it, and we would work it out. So
Yes.
and I always wondered why is this happening? This gives me a big clue here.
Yeah. So in this case, it was kind of random how they came up with their names. So the oldest child, Yongyu, was just told he needed to pick an American name.
Hmm.
And he said he wanted to be called Dick.
Okay.
And the school explained to him that that was a nickname for Richard. So he
Ah,
became Richard. Their middle son became Louis. And then when asked what Yong Qian's American name should be, Dick said it should be Roger because he liked the actor Roy Rogers, who was a
ah.
cowboy film star.
Okay. Yeah. Roy Rogers is a hero. I
Yeah.
want my little brother to be a hero. Okay.
Yes. And
Yes.
so then their Chinese names became their middle names, or
Okay.
some translation of their
Some
Chinese
some
name. Yeah.
variation. Okay. Hmm.
So Roger's name now is Roger Yanqian Tsien. Very early on, Roger got interested in chemistry, particularly in experiments that were colorful. He shared two examples in his autobiography. The first is what he called silica gardens, which is
Hmm.
where you drop a colorful metal salt into sodium silicate or liquid glass.
Yes.
And the salt forms this kind of jelly like coating, which would then grow long tendrils growing upwards. And so over
Oh.
time, you get kind of this weird, colorful, underwater garden looking thing.
Oh, interesting.
Yeah. I'll post a video on the website so that people can go and see it. It's really cool looking.
Yeah, this sounds like a fun, a little experiment for any middle schooler or even elementary schooler to do.
Yeah.
Hmm.
So he really liked it. And depending on what color salt you use, you get different colored seaweeds or tendrils growing upwards.
Yes.
The second experiment he enjoyed a lot was creating a strongly basic solution of
Hmm.
potassium permanganate, which has a very strong purple color. And when you pass this solution through a paper filter, the
Hmm.
potassium permanganate steals electrons from the cellulose in the paper, and
Yes.
this changes the way that the permanganate absorbs light. So once it
Oh.
gains an electron, potassium permanganate turns green. So you're pouring in this purple liquid and it comes out
Yes.
green on the other side. Very cool.
Ah. Oh my goodness. Oh, I wish I knew these experiments when I was in middle school. It would have been fun to do this.
Yeah. And actually, Roger, like later after he won the Nobel Prize and he was talking about these experiments that are what piqued his interest in chemistry, people are like, "Oh, we've never heard of that." And he's like, "What? You've never done this before? Let me show you." And he like demonstrated it. So he was pretty surprised that people hadn't heard of these experiments also. They're really
Yes,
cool.
yes,
And actually, potassium permanganate is really cool. I was looking into it, and it's
yes.
called the chemical chameleon because if you keep on
Oh,
giving it electrons and keep on doing these redox reactions, the potassium permanganate will turn into manganese dioxide, which has
hmm,
a brownish yellow color.
huh.
And then eventually, if you keep going, keep on doing a redox reaction, keep on giving it electrons, you'll end up with just the manganese ion, which has a very pale pink or clear color. So
Oh,
you can
wow.
transition through this whole from purple to green to yellow to pinkish. Very
This
cool.
is fabulous. Chemical chameleon.
Yes, very cool.
Yes.
In 1950, when Roger is 7, his father started working for the Radio Corporation of America, or RCA, and so the family moved to New Jersey. His parents found a new housing development that was being built in Livingston, which was perfect in terms of his dad's commute to work, and it was in a good school district for the three boys. But the developer refused to sell them a home, saying, quote, they could not permit Livingston to become a Chinatown, nor could they afford the likelihood that other customers would refuse to buy houses next to a Chinese family, end quote.
Oh, discrimination raises its ugly head.
Yes. So the Tsiens eventually had to appeal to the governor of New Jersey, Robert Minor, and he wrote a letter to the developer saying that what they were doing was illegal racial discrimination. They
Yes,
couldn't
yes.
refuse.
Yeah, I thought there are laws against it. Yeah,
Yes.
yes.
And so finally, the developers compromised by selling them a house that was completely surrounded by other houses that were already sold.
Yes, because then nobody else can
Mhm.
refuse to buy a house next to them.
Yes. Roger was a very scrawny, non athletic kid who suffered from asthma, so he spent a lot of his time indoors. But he really tried to integrate himself into American culture. He didn't take any interest in learning Chinese traditions or the language or eating their authentic foods.
Okay.
So I found that interesting.
But I can imagine that with the kind of discrimination they're facing, it's not making you unique and memorable, it's making you a target. And I can see how, as a young child, you don't want to waste your energies on that. You just want to kind of go with the flow.
Yeah, and like you were saying, like I'm sure he already stuck out because he obviously looked Chinese and so,
Yes,
you know,
yes.
but he did keep up his intense interest in chemistry, turning his basement into a lab. His dad eventually moved jobs again and started working for Exxon Research. And he would secretly bring him home chemicals and glassware or
Huh.
help him special order equipment that he needed for doing his experiments.
This is how he had access, because that was a kind of question bugging me at the back of
Question.
the head. That
Yeah.
solves one mystery for me. Okay.
In high school, he entered a summer program sponsored by the National Science Foundation, where he was assigned to work in a chemistry lab at Ohio University under
Hmm.
Professor Robert Klein. He worked on a project involving thiocyanate, which is a compound that's used to neutralize cyanide.
Okay.
So, like if we humans ingest small amounts of cyanide from certain seeds or environmental toxins, our body converts it to thiocyanate, which
Ah, I
is
see.
not toxic and can
Not
get
as
it.
toxic. Okay.
Yeah.
Mm.
So Professor Klein was interested in whether thiocyanate could bond to different types of metals at the same time. And so Roger's summer project was a lot of making different compounds out of thiocyanate and taking measurements. The compounds he made were not very well defined and his results were inconclusive.
Hmm.
But the following year, he entered a nationwide competition called the Westinghouse Science Talent Search. And as Roger says, quote, for lack of any alternatives, I wrote up my Ohio University project, trying my best to draw some conclusions from a mess of dubious data, end quote.
Been there, done that.
Yes.
This is what research is all about.
Yep.
Even now, it's not necessarily as a high schooler, even after years and years, you conduct experiments and
It's
you have
just,
to
yeah,
just look
so
at the mess of
confusing.
the data and say, oh, there is so much noise. How do I draw a clear conclusion out of this?
Yeah. And so he tried drawing some sort of conclusions in order to, because he's entering a science competition, he wants a nice story. But even Professor Klein was not really willing to back up any of the conclusions Roger drew. And Roger was like, yeah, fair enough. Like,
Yes,
this is
I
not great.
understand. Yes.
Yeah. But he entered this science competition and, he was like, not having very high hopes because he was like, oh my God, everyone else's project is so much better and cleaner than
Yes.
mine and
Yes.
so nice. And he was also very intimidated by the fact that the competition was judged by a Nobel Prize laureate, the first one in our story, Glenn Seaborg, who won the prize for discovering 10 elements on the periodic table, numbers 93 to 102, all of which are
Wow.
radioactively unstable.
Glenn Seaborg, the one who
Yes.
found 10 unstable radioactive elements. And I'll go for my water too. Hang
Yes,
on.
take a shot.
There it
Take a
is,
sip of water.
to Glenn Seaborg.
Yes. Nevertheless, Roger went on to win the entire competition to his
Mm,
intense surprise, taking home a $10,000 scholarship prize.
that is big money in those times. It probably
Huge.
funded his entire college education in those days.
It might have, or a big chunk of it.
Good
Yeah.
for him.
But the thing that gave him the most satisfaction was
Mhm.
that when he brought home the prize, the housing developers that hadn't wanted to sell their family that house started using Roger's award picture in their advertisements to show what a good school system they were part of.
Are you serious? Are
Isn't
you
that great?
serious? Hypocrisy at its best.
Yes.
Oh, this is really hilarious. They not
Yes.
only had to eat their own words, they had to
Yeah.
advertise it to the entire world to let
Yeah.
people know, oh, okay, forget that. We never said no to this wonderful, wonderful family who wanted to join our school system, who is so amazing.
Yes. Yes.
This is karma at its best. This is what karma
Right?
is about.
This
Yes,
is
this is
this is
karma.
100%. I was like, that's karma.
Yes, this is karma. Yes,
Yes.
good, very good.
Roger now graduated high school and he is facing a choice of four colleges. His choices are Columbia, MIT, Caltech, or Harvard. What to do?
Whoa,
What to
can't
do?
go wrong.
Yeah.
Well, the only thing I can say is with those kind of choices, it doesn't matter. You can close your eyes and toss a coin or spin a wheel or whatever you want, and you can't go
Yeah.
wrong.
So here's how he made his decision. His dad told him not to go to Columbia because recently there had been a lot of student protests there against the Vietnam War. And so all the unrest made him uneasy. And Roger also was like, Columbia's too close to home anyway, so that's out.
Okay.
Both of his older brothers had gone to MIT. And so
Huh.
Roger's like, well, I want to be different. I don't want to do that.
Yes, because everybody will remember, oh, you're Richard's younger brother. You don't want to be in that
Yeah,
situation,
he didn't want to
always
be
being compared.
compared.
No,
Yeah.
you're setting yourself up to be constantly compared. It's not fun.
Yeah, so MIT was out. He was very drawn to Caltech, but
Hmm.
ultimately decided against it because Nobel Prize winner Richard Feynman was no longer teaching Intro to Physics there.
Whoa. And that brought Caltech down in his eyes. Oh, wow.
Yes.
Wow.
Yeah. And so he's left with Harvard. And overall, he had a great time at Harvard. The
Hmm.
culture was great. There was a lot of diversity in both students and classes. Ironically, though, he hated his chemistry classes.
Oh, no.
And I think specifically it was organic chemistry, which
Hmm.
was, the thing that he really disliked. And I can understand that. Like
Hmm.
everyone in my major, and when I was doing biochemistry as my major, everyone was like, oh no, the dreaded OChem. We all hate OChem.
OChem, I think, gets a bad rap because people who teach it do not know how to teach it correctly. I think you faced that as well when you were learning OChem.
I had a couple of good teachers, but I had a couple of bad ones too.
However, I think you did best once you were able to get that kinetic model with which you were able to work.
Oh, yeah. I'm a very hands-on learner.
I think OChem is one of those subjects that you need to have multiple media integrated into teaching.
Yeah, and I think a lot of the professors immediately jumped to teaching us mnemonic devices to remember
Mm,
things rather than
yes.
like actually how nature works. And I'm terrible with that
Yeah,
kind
no,
of thing.
that's just memorization without understanding at all, and that doesn't work either.
Yeah, so he hated his chemistry classes and he started exploring other courses in molecular biology, quantum mechanics, astrophysics, but eventually he landed on neurobiology,
Okay.
in part because of two professors, Professor David Hubel and Torsten Weisel,
Hmm.
who both of them would go on to share the Nobel Prize in 1981, along with Dr. Roger Sperry, for understanding how light signals from our eyes are processed into visual information in the brain.
Wow. Is it enough to
Yes.
just take a sip of water?
Yes.
Do we have to
Three
genuflect
small
also?
sips, maybe.
Should
Yeah.
we also do some genuflecting here? I don't know.
Yes. Yes. So these two Nobel Prize laureates who would eventually be, like at the time that they were lecturing and teaching Roger, they hadn't won the Nobel Prize yet, but um, they were just so enamored by neuroscience and the way that they lectured to their students, saying like neuroscience is the next frontier and we need to understand how the brain works. And Roger was just completely taken in by that. And he
Yeah.
was like, yes, that
Makes
sounds
sense. If they
fascinating.
went on to win the Nobel Prize, obviously they were highly passionate about it, and they were able
Yes.
to communicate their passion for their subject to their students and basically ignite the same passion in their students as well.
Yes. So Roger decided, I'm going to go into neuroscience. So he asked Professor Hubel if he could join his lab, but he wasn't taking on any undergraduate students. And so he suggested that Roger apply to the lab of Dr. Nelson Kiang instead, who worked on auditory neurophysiology, or how
Mhm.
sound signals from the ear go to the brain and are processed. So Roger did that. And then for graduate school, he wanted to continue working in neuroscience. So he asked Professor Hubel and Professor Kiang which were the best schools for neuroscience. And they told him Cambridge or Cambridge. So either stay at
Oh,
Harvard in Cambridge, Massachusetts, or go to Cambridge in the UK.
cool. What a nice way to put it.
And so Roger was like, oh, well, I'm up for a new adventure. Let's go to Cambridge in the UK because I've done Harvard. Let's go see something new.
See what something else has to offer. Yeah.
So he applied for the graduate program there and he got in. And he was told that he would be working under Dr. Richard Adrian, whom he had never heard of. So he called his older brother Dick, who was now a professor of cardiac electrophysiology at Yale. Yeah.
Wow.
And Dick said, oh, yeah, I know Professor Adrian. He is
Okay.
the son of Dr. Edgar Adrian, who is a Nobel Prize laureate.
Wow. He is in such a rarefied atmosphere.
Yes,
This, how often does this happen to anybody in the
I
world?
know.
Just
I
wherever
was
he
not
goes,
kidding about this. This is
yeah,
like,
wherever he goes, he's
yeah,
surrounded by these extremely knowledgeable, passionate scientists.
yes.
Amazing.
So,
We forgot our sip.
yeah, take a sip. So, Dr. Edgar Adrian won the Nobel Prize for understanding the electrical activity in the brain. And so Dick told Roger that Richard Adrian, the son, was a skeletal muscle electrophysiologist.
Yes.
And Roger was like, oh no, I don't want to study muscles. Muscles are backwater research. I wanted to work on the brain.
Okay.
But Dick told him he would be okay. Professor Adrian would probably let Roger choose his own thesis topic, so
Yeah.
he could probably work on the brain if he wanted to. So Roger decided to take a chance, and he went to Cambridge. And when he first met Professor Adrian asked Roger, are you Roger Tsien? And Roger was surprised that Adrian had pronounced his last name very correctly.
Yeah.
Professor Adrian then asked if it was true that Roger thought that muscle research was a backwater
No.
field
Wait.
of research.
Wait. How did he find out? How did he find out?
And Roger realized that his brother Dick had met with Professor Adrian over the summer and
His
spilled the
brother
beans.
sold him out?
Yes.
That is so mean.
That's such an older brother thing to do, I feel
That
like.
is so terrible to set
Yeah.
him up in his brand new place where he's going. He doesn't know anybody. He's leaving one country to go to another, and his brother sets him up with his new professor. I
Yes.
would want to, I would want to pound him into the sand
Yeah.
when I came back home for
I would want to
Christmas.
kill him. Yeah.
I absolutely would.
Right? But Professor Adrian was very gracious about it.
Oh, of course, I mean, I'm sure that's why the brother was able to open up as well. I get that. I totally get it. I'm
Yeah.
sure
I think the older
his,
brother was just kind of being mischievous and was
yes,
teasing Professor
yeah,
Adrian, like,
yeah,
hey,
they're probably
yeah,
the
they're
two
probably
older
good friends.
guys, they're
Yeah.
probably rolling their eyes at this young
Yeah.
wannabe scientist who is like,
Yes.
"Ew, muscle research is so ew, I want to
Yeah.
be in the brain." And they're probably rolling their eyes and making fun of him,
Yeah.
like typical older brothers and friends do. But
Yes.
not fair,
Not
not
nice.
fair.
Yes. But Professor Adrian was gracious about it, and he confirmed that he would even let Roger transfer to a neurophysiology advisor for his thesis if he wanted to. But Roger quickly realized that actually neuroscience research meant doing a lot of electrophysiology work, which he did not
Mhm.
enjoy. So basically, when a neuron gets excited enough, sodium channels on the membrane of the neuron open up,
Mhm.
allowing a huge influx of electrically charged sodium ions into the neuron, which creates an electric pulse called an action potential. The action potential starts in the main cell body of the neuron and then travels down the axon and ends up at the axon terminal where the neuron forms synapses with other neurons.
And thus the signal gets transmitted.
Yes. Here in the axon terminal, the action potential causes calcium channels to open up and calcium ions enter the neuron. And this signals a release of neurotransmitters that go and signal other neurons or other parts of the body.
Okay.
And so the way that Hubel and Tsien at Harvard were doing neuroscience was by putting a microelectrode into the brain of the animal, and then they would record the activity of an individual neuron. So they would give the animal some sensory stimulus and then
Hmm.
see if that neuron, that individual neuron that they were measuring, lit up in response to
Got
the
it.
sensory
Yeah. So
stimulus.
you could hone in on your most likely neuron up in the brain
Mhm.
and give a signal, let us say, on the foot or the finger or something like that,
Yeah.
or
Well, they
create
were
some
doing
sort of a,
visual things, so they would probably give
okay.
the animal something to look at. Yeah.
Yes, give something to look at and see if the neuron lights up. But it could potentially be the wrong neuron that you're chasing. And so you
Yes.
have to go and do the same thing again with your next neuron now, and
Mm-hmm.
so on and so forth. Yeah, that
Yeah,
okay,
and you had to
that
do
makes
that
sense.
like a bunch of times until you came up with a pattern or some network that made sense.
That's right. Okay.
And Roger was like, that is so tedious. That's so
Absolutely.
slow.
Yeah.
Yeah, we know that the brain is super complex, trillions of neurons all firing and signaling each other. And so he thought, what's a better way to do this? And he said, quote, ideally, one would stain the neuron with a dye that would visibly light up or change color whenever and wherever a neuron fired an action potential, end quote.
That's
Yes.
the idea coming through. Okay.
Yeah.
Yes.
So in 1972, in graduate school, he decided to try and design a dye specifically to see neuronal activity. He thought the best way to do this would be to either create a dye that would become visible when the sodium channels opened up when the neuron initially got excited, or maybe a dye that responded to the electrical changes that happened across the neuron's membrane.
Got it. So the dye is inert, but then it lights up because of its interaction with the sodium channel, or because now it is being stimulated by the electric impulse.
Exactly. Something like that is what he's thinking. But Either way, in order to do this, that meant he had to study organic chemistry, which were
Back
the
to
classes
organic
that,
chemistry, which he hated it for probably good reason. Okay.
yes. But he was very determined, and so he found a mentor, Dr. Ian Baxter, in the chemistry department to help him. And he realized that actually, if he was studying organic chemistry for his own purposes, he enjoyed it a lot. So kind of
Hmm.
like back to what we were saying, like
That's right.
if you teach it the right way, you give a student the proper motivation, they will learn.
I think that's really what's missing sometimes with these complex teachings. You can't just go on droning about something, draw some structures on a blackboard, and expect everybody to pick it up.
Yeah. So, although he was able to create molecules that did bind to sodium channels, he wasn't having much success with them acting as an indicator of whether the neuron was getting excited or not. But then he read about a new dye, arsenazo 3, that was able
Okay,
to bind to heavy metals and calcium. And he was like, oh, hey, that could work too, because once the action potential reaches the terminal, calcium ions come in and trigger
that's
the
right.
release of the neurotransmitters.
Yes,
So maybe targeting calcium is the better way to go.
that makes sense because the interaction with sodium is not creating that different signal that you're looking for.
Mm-hmm. Yeah. And
Hmm.
so maybe time to switch targets. And actually, there are plenty of dyes, hundreds of dyes that could bind to calcium. The problem was, though, that inside the cells, these dyes would also bind to magnesium ions, which were about 4 times higher in concentration than calcium
Yes.
ions.
Okay.
And so what he did was very clever. He found another molecule called EGTA, which was a colorless buffer,
Yes.
but it had selectivity for calcium, meaning that
Yeah.
it strongly preferred to bind to calcium rather than magnesium.
Yes.
And then with a bit of playing around, he found a way to kind of combine EGTA's selectivity with a dye to make a new molecule called BAPTA.
Ah, Bacta is well used today.
Yes.
Wow. I didn't realize that's how it came about.
So his version of it was a bit rudimentary, and later other
Hmm.
scientists took that concept and refined it to make other versions, and that's probably actually what we use today. But that
Yeah.
initial concept, it was the first dye that could selectively bind to calcium inside a cell and
Yes,
therefore tell you whether a neuron was excited or not. So it was
that's right.
huge.
Yes. Yes, that I can see this.
Mm-hmm.
Wow. Exciting.
Hey, Rum, do you know what's going on back there?
Biology working?
Biology working. That's basically the whole show.
We're the co-hosts of the Grow Everything podcast, where we talk to scientists, founders, and engineers who are using living organisms to make everything: materials, medicines, and food.
But you forgot beer.
Of course, beer. New episodes every week.
Subscribe wherever you listen.
Hi everyone, Aarati here. I hope you're enjoying the podcast. If so, and you wish someone would tell your science story, I founded a science communications company called SciComm, that's SYKOM, that can help. SciComm blends creativity with scientific accuracy to create all types of science communications content, including explainer videos, slide presentations, science writing, and more. We work with academic researchers, tech companies, nonprofits, or really any scientist to help simplify your science. Check us out at scicommer.com. That's SYKOMMER.com. Okay, back to the story. So that was basically his PhD project. He did that for his PhD. And then he stayed at Cambridge for his postdoc. And during this time, he started collaborating with another new faculty member in the physiology department, Dr. Timothy Rink, who was
Hmm.
also interested in making calcium selective electrodes.
Okay.
They became really good friends, and Tim would invite Roger over to his house and, invite him to parties where his wife was there. And in 1973, at a Christmas party, Roger met Tim's sister-in-law, Wendy Globe, and they hit it off immediately.
Hmm.
They started dating kind of secretly at first, and then several months later, when Tim and his wife found out, they were shocked at how well they had managed to set up these two without even meaning to.
Meaning to.
Yeah.
Inadvertent matchmakers.
Yes. Yeah. So after his postdoc fellowship was up, he tried his best to get a job in Britain because he wanted to stay close to Wendy. But finding a research position at the time was very difficult. So he turned back to the US.
Okay.
But there also, he had a hard time. And the reason was kind of interesting. He was an interdisciplinary scientist with expertise in both biology and chemistry, which
Yes.
was weird for the time. So
They are unicorns today who are highly
Yes.
prized.
yes.
People recognize interdisciplinary is the way to go and break down those boundaries of science.
Yes. But back then, biology departments were rejecting him because he was too much of a chemist, and chemistry departments were rejecting him for being too much of a biologist. So,
They wanted purists.
yeah,
That's not how science works anymore.
he was way ahead of his time. Finally, though, through some network connections, he was able to land a professorship at Berkeley. But his startup funding package was extremely low, just a few $1,000s. So to set up his lab, he had to rely on old equipment or create some very strong justification for getting new things.
Okay.
Another professor who worked in a lab next door donated an old fume hood to him. And so for the next 7 years, all the chemical reactions that were done in the lab had to be done in that single donated fume hood. But still, he was able to make improvements on his calcium ion indicators and synthesized a sodium ion indicator. So back to the sodium channels.
Back to sodium. Okay.
Yep. And take images of both of these indicators inside individual living cells. And in 1982, he married Wendy, and so she came to the US to live with him.
Okay.
But after several years, he outgrew his lab at Berkeley.
Okay.
Not only was he constantly struggling with limited and outdated equipment, but he also felt like his research was being boxed into just imaging ions, like just looking at calcium ions or sodium ions. And he wanted to look at bigger biochemical molecules and their interactions.
Yes.
He was particularly interested in cyclic AMP, which acts as a messenger inside cells.
Yes.
So once a signal comes from outside of the cell, and that signal could be either a neurotransmitter or a hormone from some other stimuli, cyclic AMP runs around inside the cell, activating pathways that regulate the cell's response.
Yes.
And these could be pathways that are metabolic, gene expression pathways, enzymatic pathways, all sorts of things.
Okay.
And actually, in 1971, Dr. Earl Sutherland had won the Nobel Prize for discovering that cyclic AMP worked as a messenger inside the cell as a response to epinephrine. So it's a hugely important molecule.
We have to take a sit now.
Yes, because I had to mention.
Sutherland.
And so in 1989, Roger and Wendy moved to UC San Diego, where he was able to get a much bigger lab, and he was able to, pursue this line of inquiry.
Yes.
At UC San Diego, there also happened to be another scientist named Susan Taylor, and she was an expert on cyclic AMP and its role in activating protein kinases.
Okay.
So really, how cyclic AMP works is once a signal is detected from outside the cell, cyclic AMP goes and binds to a protein kinase, usually protein kinase A.
Okay. And protein kinases are the ones who actually move the phosphate around on the various molecules to either activate or inactivate them.
Yes. So protein kinase actually is made up of two smaller subunits. So once cyclic AMP comes, it binds to one of the subunits of protein kinase, but the other subunit goes and starts phosphorylating things and dephosphorylating things and
Yes.
turning on and off enzymes, altering gene expression, metabolism,
Yes.
all of that. So cyclic AMP is just acting as the secondary messenger that once the cell senses something, cyclic AMP goes and says to protein kinase, hey, the cell has sensed something.
It's really like a relay race. Like out
Mm-hmm.
there, all the way out there came a signal, a hormone or something. And somebody on the cell surface got tickled. And that tickle then goes in and is transmitted over to the cyclic AMP, who is very agile, and he goes running around, bounding away, goes to the protein kinase, which is more internal to the cell. And now it's like the protein kinase is now, oh, I need to activate X pathway or Y pathway. And the
Yeah.
way the pathways get activated or deactivated is through the phosphorus that is attached or removed. So that's how it's working.
Yes. And so Roger comes up with a really cool idea, now knowing this about how protein kinase and cyclic AMP interact. He thought instead of trying to add a dye or indicator to cyclic AMP, what if we add an indicator to each subunit of the protein kinase?
Uh-huh.
So he added a green indicator called fluorescent to one subunit and a reddish dye called rhodamine to the other.
Yes.
And so now when you look at the protein kinase, if it's intact, if both subunits are together, because the green fluorescent and the red rhodamine are so close to each other in space, you get a phenomenon called fluorescence resonance energy transfer, or FRET.
Mm-hmm.
So any light that's coming from the green fluorescent gets absorbed by the rhodamine and is emitted as red light. So when the two pieces are together, you won't see any green light because it's all being emitted as red light from the rhodamine.
Okay, due to the FRET phenomenon. Yes.
Yes.
Mm-hmm.
But when cyclic AMP comes along and breaks apart the protein kinase, then the two dye indicators will separate from each other because the subunits have separated.
Yes.
And so now you will see both red and green light in different places in the cell.
Got it.
And so this not only tells you whether the protein kinase has been activated by cyclic AMP or not,
Yes.
but by measuring the ratio of red to green, you can also tell how much of the protein kinase has been activated.
It is quantifiable. Okay.
Mm-hmm.
Yes.
And so, although this was great, they got some good papers out of this, they were able to publish. This process still had its drawbacks. It was very labor intensive. You had to purify the two subunits of protein kinase so that you could attach the dyes in a way that did not destroy the function of the subunits, and then inject those two subunits back into living cells.
Yes.
And so Roger was like, we need to get away from using dyes if possible. They're such a pain. Instead, what if we could fuse the proteins we're interested in with a naturally occurring fluorescent protein? That would be ideal.
Yes. We are inching closer to where we are today. I can see it. Okay.
Yes. And so he started turning to examples of naturally occurring fluorescent proteins in nature, and mostly he was looking at bioluminescent algae, jellyfish, fireflies, fungi. Roger's like looking at all of these and studying how their bioluminescence came about, what proteins were causing their bioluminescence, and how could we co-opt these for our own research purposes.
Absolutely. Mm-hmm.
Then in 1992, he came across a paper from Dr. Douglas Prasher at the Woods Hole Oceanographic Institution in Massachusetts. Dr. Prasher had successfully been able to clone a bioluminescent protein from the Aquaria victoria jellyfish called green fluorescent protein, or GFP.
I was biting my tongue to not say GFP because I knew I would preempt you. So I did not want to say it. Don't say GFP.
Yes.
Green fluorescence protein. How many beautiful images they have created with that GFP. Yes.
Yes. So GFP had first been isolated and studied back in the 1960s by Dr. Osamu Shimomura. But no one had looked at whether it could be used in other animals or cells. So then in the late 80s, Dr. Prasher wanted to take up the study of GFP, but the NIH and NSF both declined giving him grant funding. Isn't that crazy?
Sounds crazy today.
Yes.
It's like one of the fundamental backbones of research today.
Yes.
It's so cost effective for ongoing research to use GFP. You have the sequence in every lab, every cloning lab has the GFP sequence ready to go to tag on to anything that people wanted to tag on to.
Yeah.
And the highest institutions of science in America are like, nah, this is not
No.
interesting enough. I don't want to fund it. It reminds me of that lady from Alaska, Sarah Palin, who was scoffing at research on fruit flies. She was scoffing and making fun of it. Who wants to study fruit flies?
Yeah.
Without the least, it was obvious she did not understand that it was the model organism on which we could base a lot of human complexity could be decoded
And
using
that's
these.
bad enough. I mean, she's a politician, which
Yes.
doesn't give her a pass at all, but at least she was a politician. This is like the NIH and the NSF saying this GFP is not worth researching. And
Incredible,
it sounds
incredible. It's
just crazy to hear that today.
Yes, yes. I'm glad it was documented somewhere that they rejected these grants. From the way you have set up the story, it is pretty obvious that this is going towards a Nobel Prize. And
Mhm.
so I'm glad that the naysayers were documented as being naysayers.
Yes, I think it's important to point it out. Yeah.
Yes, it's like
Yeah.
you made a foolish decision before. I cannot trust you anymore.
So the people who are the heroes of the story, who did give him funding, are surprisingly the American Cancer Society. They gave
Mm,
him a small grant, and using that funding, Dr. Prasher was able to clone GFP, the GFP gene.
yes.
So essentially, he was able to make copies of the gene and able to isolate almost the complete DNA sequence.
Hmm.
Today, that would be, I think, like figure 1A of a paper, maybe.
Or
But
even in the supplemental, yeah, correct.
yeah,
But that was
yeah.
that
But
was the
back then,
groundbreaking.
yeah, that was
groundbreaking.
groundbreaking. That was
Correct.
huge with the technology limitations
Mhm.
that they had. Huge
Yeah.
achievement.
Even to actually sequester out the DNA, the techniques were so bad. It was all radioactive based in those days.
So Dr. Prasher was able to write a paper on this and also suggested in that paper that GFP could be used as a marker for other molecules.
Yes.
But before he could take his work much further, his funding ran out. Around this time, Roger read this paper and reached out to Dr. Prasher,
Mhm.
as did another scientist, Martin Chalfie.
Mhm.
Martin Chalfie was interested in putting GFP in different animals, and he was successful in expressing GFP in E. coli bacteria and in C. elegans, my
Yes.
grad school model
Your
organism.
grad school work. Yes.
Yes, my
Yes.
little worm.
Yes.
And so Martin Chalfie was able to show that GFP could in fact work as a fluorescent tag in living animals. Roger, on the other hand, was more interested in expanding the toolkit. Remember, his work with protein kinase and cyclic AMP had required two colors. He had
Correct.
the green fluorescein and the red rhodamine dye.
So now he needs a red guy.
Yeah, so GFP gives us green, and Roger's like, let's make more colors.
Yes.
In order to do that, he first had to understand how GFP worked in the first place.
Hmm.
A lot of the other bioluminescent proteins that they had found in nature from other organisms
Mhm.
usually required some other enzyme or cofactor to actually fluoresce.
Okay.
And that's what kind of made them bad for science, because then you
Okay.
needed to have like two things that you're putting
Yet
into the
another,
cell.
yet another component. Yeah, it becomes
Yeah.
the system becomes more and more complex. You're already adding these fluorescent proteins to understand a complex system with various moving parts. And you're hoping just by adding one tag, you can decode it. But now, in order to decode the tag, you need another decoder. So
Yes.
it becomes even more complex. So you need to keep at least the tags fairly simple.
Yes. But Roger found that GFP was special because it worked all on its own as long as oxygen was present. If
Yes.
oxygen
Okay.
was there, you could get this oxidation reaction that would make the GFP glow.
Okay.
He also looked at the crystal structure of GFP and found that when it folded, it formed a special structure that almost looks like a cylindrical lantern with a glowing
Oh,
light on the inside. That glowing part on the inside is called a chromophore, and it absorbs blue light and emits green light.
yes.
And then the rest of the protein structure of GFP that forms the cylindrical lantern protects that light from water or any other unwanted chemical reactions.
So pretty.
Yes, it's really cool. And so now, because he had spent so much time understanding the structure of GFP, he and his lab could start to make small modifications to the DNA sequence and start to make alternate forms of GFP that either fluoresced at different wavelengths. So, for example, they were able to make YFP, yellow fluorescent protein,
Yes, yes.
BFP, so blue, or CFP, cyan. So they were just able to shift the green
The
wavelength.
wavelengths at which, the activation and the emission wavelengths have changed a bit.
Yeah, so he was able to shift it a little bit left or right to make it yellow or blue. And they were also able to create brighter versions of GFP that gave a stronger fluorescent signal than the wild type. Roger and his lab tried for some time to get a red fluorescent protein, but they couldn't figure out how to do it. They couldn't shift the emitted wavelength that far away from green. That is until another scientist, Dr. Sergei Lukyanov's lab at the Russian Academy of Science, got involved. They discovered a species of red coral called Discosoma that got its red color from a homologue of GFP.
Another brother.
Yeah, and no one had looked at the coral before because the coral itself wasn't bioluminescent. But it turned out that the protein that makes it red is a fluorescent protein. And so finally now Roger and his lab were basically able to look at nature's cheat code and see how did nature do it. And it turns out the red fluorescent protein, which was called DS red, DS for Discosoma, it had a chromophore that looked exactly like GFP, but with an extra structure attached. And they were now able to look at that structure and study that structure and see.
Yeah. And now you add that to GFP and make
and make it shift to red. Yes.
Amazing. Nature, the lesson
Can't
stays
beat
the
it.
same. Can't beat nature. Learn from
Yes.
nature, observe nature.
And that's actually what Roger was saying too. He was like, none of this would have been possible without the jellyfish. People are all saying, oh my gosh, you're so amazing. You won the Nobel Prize. And he's like, the jellyfish is the one who did it.
Yeah, it's the jellyfish who is amazing. Nature, that's amazing. I just borrowed her cheat sheet.
Yeah, exactly. So now that they've figured out DS red, again, they're able to start making mutations and they start making slightly different reds. They got like a more orangish red, a deeper maroon red, a scarlet, all these various shades of red. But what can you name them all? You can't name them all RFP. They're all slightly different.
Yes.
And so that's when they started getting creative with the names. And that's how we get the names M cherry, M strawberry, M tomato, and M raspberry. There's they have all different fruits.
I have worked with so many of these. And it's only now that I know how the names derived. Because until now, I have only focused on what my needs are and how different I need the colors to be. And what are the filters, excitation and emission filters that I have available to me for my experiment, and then choose the appropriate color based on that.
So he created this whole rainbow of colors, basically.
Nice.
And by the way, if you're wondering why it's M cherry and M raspberry, the M stands for monomeric. The original DS red in the coral formed tetramers, which would have messed up our experiments. And so Roger's lab did a lot of work getting the proteins to work as monomers.
That's a lot of work they have put in.
Yes.
So many steps.
Oh my gosh. I am skating over so much of his work. I am skating over so much.
Even every step that he's taking, I can see the amount of effort. Like multiple people have to spend years making it happen. We are just able to say it like click, 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 one sentence here and two sentences there. But no, this is intense failure, and no, that didn't work, and let's try this and make this happen and change this and change this. and
Yeah.
until you finally get where you are, and the perseverance that is required for that.
Yeah, and I was listening to his Nobel Prize speech, and in that, when like he's describing his work, it's just incredible the amount of work and the depth of knowledge he has. He was like,
Hmm.
"Oh, we mutated this serine to a threonine, and then that didn't work, and so we then went back and we're like, oh, but the serine is important, so why don't we try making a glycine instead?" And he's like got the whole amino
Hmm.
acid sequence down, the whole DNA sequence down. He knows exactly how things fold and which
Yes.
amino acids interact with each other, and if you change that one you can't change it, otherwise it screws up everything, but you could do whatever you want to that other one, so we were messing around with that other one. And I was just like, oh my
Wow.
god, this is
Yes.
just you know.
Just the ultimate master who knows exactly how to move his little chess pieces.
Yes,
Beautiful.
so really
Beautiful.
amazing. I'm skating over so much. I'm like, just summarizing so much work.
Yes, otherwise we'd be here for the next 3 episodes
Yeah.
just talking
For
about
real.
this person.
And so finally he's like achieved his dream. He's got red, he's got yellow, green, blue, and now scientists could use all of these colors to make these beautiful scientific, really enlightening images that show
Yes.
us very precisely different structures, which proteins are interacting with each other, where cells are located, all these sorts of things. In 2008, Roger got woken up by a call in the middle of the night from the Royal Swedish Academy saying that he had won the Nobel Prize for his work on fluorescent proteins.
Wow.
He shared the prize with Dr. Osamu Shimomura.
The one who actually found the GFP to begin with.
Yes, all the way back in the
Mm-hmm.
60s.
Mm-hmm.
And then Dr. Martin Chalfie, who was the one who tagged it in bacteria and C. elegans,
Oh.
who used GFP to tag proteins. Yeah.
Oh, okay. So the one who actually isolated it, Prasher, didn't get included in this?
Correct. And so that was actually a bit of a controversy for this Nobel
Hmm.
Prize. He was the one who cloned GFP and was left out. The prize can only be shared by up to 3 people. And after his funding had run out, after he had cloned GFP,
Mm-hmm.
Douglas Prasher had struggled to find a job in science, and he actually ran out of his savings, and he ended up leaving science altogether and becoming a shuttle bus driver for a Toyota dealership. So that's what he was doing when he heard about the Nobel Prize being awarded in GFP.
Oh, that's heartbreaking. I'm heartbroken.
He said he was very happy for the winners, although he was somewhat surprised that the fluorescent protein from a jellyfish ended up being that important that it went on to
Hmm.
win the Nobel Prize. All 3 of the Nobel Prize winners thanked Douglas Prasher profusely in their Nobel Prize speeches, saying that his work had been invaluable. And Martin Chalfie even said they really should have given his spot to Douglas Prasher and just left him out of it entirely.
Actually, that is my sentiment as well, although I am not the right person to talk about these kind of things. I'm speaking too big for my boots. But yeah,
Yeah, like, I don't
I
know
think
how they made
Prasher,
these decisions, but yeah.
correct. Yeah. I think the fact that he had to give up science played a role there. The committee might have found it, though he's divorced from science now, so let's leave
Yeah,
him alone, or kind of thing. That's not fair.
yeah, maybe.
Very difficult. Very
It might have
difficult.
been. I don't know. Roger invited Douglas Prasher to the Nobel ceremony as his personal guest and paid for all of his expenses to travel there and stay there. Roger also invited Douglas to join his lab at UC San Diego, and Douglas finally accepted that offer in 2012.
Okay, scientists looking out for each other to the
Trying,
best of
yeah.
their ability.
Roger continued developing fluorescent proteins, even making an infrared fluorescent protein. He started collaborating with medical scientists to find uses for his proteins, like using them to light up cancer cells so that surgeons could more easily remove tumors,
Yes.
and using them to understand long-term memory storage in the brain.
Hmm.
He also found that fluorescent proteins, surprise, surprise, were a great way to get high schoolers interested in science because they got to play with pretty bright colors.
Yes. Who doesn't like to play with colors?
Yeah.
Cue young Roger who played with the potassium permanganate.
Exactly. That's what got him into science in the first
Yes,
place, like
yes.
seeing all the beautiful colors, watching them. Yeah, and so things really came full circle. Roger started getting involved with outreach programs and student lunches with students who were interested in science. Sadly, in 2016, Roger died suddenly while biking on a trail in Eugene, Oregon.
Oh,
By this time, he was a part-time lecturer at UCSD, but he was
mm,
spending time doing a sabbatical at the University of Oregon.
okay.
He was only 64, and so his death was very unexpected. Although his family did not disclose the exact cause of death, it may be important to note that he had survived a stroke and a battle with prostate cancer a few years earlier.
Okay. So there had been some problems beforehand. Okay.
Yes, and the fact that he died while biking, I'm assuming a strenuous trail, it sounded like he was maybe pushing himself really hard that day or something, and,
Could be.
maybe something gave out. Not
Yeah.
sure. Regardless, he was taken from us too soon, but there's no doubt that he absolutely changed the scientific world. And if you go to Wikipedia and you look at the sheer number of awards that he got, starting all the way back in high school with him winning that $10,000 scholarship prize,
Yes. Yes.
throughout his life, it's just like this huge list of awards that he's
Yes.
won. And yeah, that's the story of Roger Tsien.
Amazing. And before we forget, we should take a sip in his name as well and for
Yes,
all the three scientists who
all the
actually
three
won the prize.
scientists. Yes. Salud.
More power to science. We need more people like Roger, and we need more people like Prasher also.
Yeah, some of these people are just really amazing and really big people, like it doesn't seem like he harbored any bitterness really at all, or not that he showed to the outside world. He was
Hmm.
always just very happy, when Martin Chalfie and Roger Tsien contacted him about his 1992 paper saying, hey, we read your paper, we want to study GFP some more. He gave them everything. He was like, here's all the information I have. Here's some samples that I created. Here's, some protein that I've isolated. Take it
So
all.
generous.
Yeah, do everything, do what you need to
So
do.
Chalfie and Roger basically built their work on his shoulders,
Mm-hmm.
and
Yes.
yet he didn't get recognized.
Yeah. And they were fully cognizant of that. Both Roger and
Yeah.
Martin Chalfie were like, he really deserves to be up here with us.
Yes.
Yeah.
Wow. Amazing, amazing story. Thank
That
you
you
for
probably
sharing.
never knew about GFP, right? Like, who
I
knew
did not
all of this
know,
history
I
behind
did
it?
not know this much information. Like I said, I'm so focused on how to make this work that I don't really often have time to deep dive into who might have thought of making this happen.
Pretty amazing story, I thought. So,
Yes,
yeah.
awesome. Thank you so much. Beautiful story.
Thanks for listening. If you have a suggestion for a story we should cover or thoughts you want to share about an episode, reach out to us at smartteapodcast.com. You can follow us on Instagram, TikTok, and Blue Sky at Smart Tea Podcast, and listen to us on Spotify, Apple Podcasts, YouTube, or wherever you get your podcasts. And leave us a rating or comment. It helps us grow. New episodes are released every other Wednesday. See you next time.